my boots on and lace them up. Hard work, not another day's work.
So I'll go over what cognitive neuroscience is. So I'll give you a little bit of history of cognitive neuroscience and how that comes from many other fields, psychology and neuroscience, a combination of those. I'll talk about some of the current questions in cognitive neuroscience at a very, very broad level. So realize that when I say this, there's many researchers doing lots of research in each of these areas. I'm going to just give you the broad overview of each of those areas. Then I'll talk a lot about some of the methods in cognitive neuroscience. There are many methods. I'll focus on the two that I use in my research and are probably the most popular among cognitive neuroscientists, but there are other methods as well. And then I'll talk about some future directions of cognitive neuroscience. Now, this is partially driven by myself in thinking what I think the future of cognitive neuroscience is, as well as I just submitted an NIH grant 3 days ago, so that's sort of what NIH is really pushing at this point. So that's sort of what they think the future of cognitive neuroscience is. And so if you want funding, you also have to think that your research is going in that direction. So I'll talk about some of those in the context of what has been studied and sort of where we think that the field of cognitive neuroscience is going. And then, as you can see in my outline, I don't have any specific topics related to library access and how librarians can assist in my research and in my students' research, but I'm happy to, of course, address those questions. I just didn't know how to actually put it in the talk, but I'm happy to discuss that and get questions from everybody here. So what is cognitive neuroscience? So it's a very broad area. So it's really about the study of how we think at multiple different levels. So our mental processes are really driven by what's happening in the brain. At least if you have the philosophy that I have, is that everything is determined by what's happening in the brain. And so in order to understand those mental processes at that level, we have to study what's going on in the brain. So it's combining psychology and then constraining those psychological theories with what's actually happening in the physiological processes that are going on in the brain. So really, we think that all complex psychological phenomena are really happening within the brain and can be, our understanding of what's happening at a psychological level can be enhanced or constrained by what's happening within the brain. And then it also, of course, has many applications to mental illness as well. So what I'm going to talk about is really focusing on understanding mental processes in normal functioning brains, but there's also application to what goes wrong. Of course, if you understand what's happening in the normal functioning brain, you can also understand what's happening when something goes wrong in the brain. Of course, there's also people that are considered developmental cognitive neuroscientists. They study the development of cognition and the neural development over the time course, which is not only children, it's also elderly, so you got both ends of the time course. My research, and what I will mostly focus on today, is really a normal, healthy brain developing in realistically in college students, because that's who we study primarily. But realize that there's a lot of other fields within cognitive neuroscience that I won't talk too much about, but there are people studying mental illness as well as the development and changes that happen within the brain over the course of the life. And of course, all of what I'm going to talk about has really applications to many other fields. So it's both a combination of these different fields as well in the development of cognitive neuroscience, but also current research in cognitive neuroscience has applications and interacts with many of these other fields. So specifically cognitive science, so these tend to be people who are in either a psychology program or in some universities have an actual cognitive science program that are separate program, or sort of an interdisciplinary program that include people from computer science, any sort of linguistic sciences, and psychology generally. As you heard, my PhD is technically a joint degree in neuroscience, psychology, and cognitive science because we didn't have one specific cognitive neuroscience degree, so it's sort of a combination of all of these fields. It also relates very heavily, or cognitive neuroscience depends on models that have been developed over the years in cognitive psychology and has application to that field, as well as clinical psychology, so what we learn about the normal human brain can be applied in clinical settings as well. And then a lot of our models, cognitive models, are constrained by what we understand from the neuroscience research. And then, of course, can also be applied to neurology. So clinical application in a different respect. So there's certain disorders are sort of under the realm of clinical psychology, and other ones, brain damage. If you get in a car accident, you lose part of your brain, then that would be considered part of this neurology. So I'll talk about research in kind of all of these different areas, but cognitive neuroscience is really an interdisciplinary field. It sort of connects to all of these other fields. And so sometimes it's actually kind of a challenge for me is that I don't really I feel kind of out of place everywhere I go. So I'm not really a psychologist, and I'm not really a neuroscientist. I kind of am stuck in between, and so that can be a little challenging. So, so the history of cognitive neuroscience, there's a long, long history. I'm going to go through it in about 3 minutes, so I'm going to skip over a lot here. But there's a large influence of ancient philosophy on the development of cognitive neuroscience. There's sort of basic ideas that have come from philosophers. And then we'll talk about a few cognitive experiments. Now, just remember, these cognitive experiments weren't there was no field of cognitive psychology or cognitive neuroscience at the time these experiments were done. It was just sort of people testing some ideas, and then those sort of experimental results have developed into these broader fields over the years. And then we'll talk about the cognitive revolution. So, 1950s, 1960s, computers became, a very hot topic. People wanted to know how the brain computed, and they used the analogy of a computer for doing this. So those kind of models are still very prevalent within the field of cognitive psychology specifically, and then those models are also applicable to cognitive neuroscience. And then we'll talk about how we've taken sort of this history of cognitive psychology, and then what do we do with neuroscience? How do we sort of fit that together with what we know from cognitive psychology? So that's kind of the newer area of combining these two fields. They've sort of evolved. You can think of them as evolving sort of independently, and then at some point somebody said, oh, well, actually what we learn from cognitive psychology can inform neuroscience research, and what we learn from neuroscience can sort of constrain the models of cognitive psychology. So I'm only going to mention a few, but there's a lot. I mean, if you talk to a philosopher, I'm sure they can give you lots and lots of history of the philosophy of mind and philosophy of science. But these ideas about thinking about mental function come from, ancient philosophy. So fundamental questions about the nature of the mind. So for example, the idea of tabula rasa from Aristotle, or I think therefore cogito ergo sum, I think therefore I am. So these are sort of the ancient philosophers were even talking about, theorizing about how the mind works. Now, of course, we've developed into these different fields of study, but really it goes all the way back to these ancient times where people are really starting to think of, what's going on? What's causing us to, think and behave the way that we do? And then, we're jumping ahead quite a while until once people started doing actual experimental research in the area of cognitive psychology. So this is one of the early experiments that were done specifically on memory. So I should say my research is on memory. So many of my examples are going to be focused on memory. So this is Ebbinghaus's experiment that was done in 1885. I'm sure all of you can relate to this. If you study something now, if I test you in 2 minutes, you can probably remember that. If I test you tomorrow, you're not going to remember as much information. If I test you 3 weeks from now, you're not going to remember much information. And that learning curve goes down. So over time, your ability to retain information decreases. So kind of don't worry about the details of this, but it's basically just saying if you study a list of words and I test you immediately, you're going to be able to retain 100% of that information. Over time, you're going to lose the amount of that information. Although you still do, even if I tested you 31 days later, you're going to feel like your memory is really poor, but you're going to remember a little bit of it. It's surprising sometimes what you remember when you don't sort of plan to remember that information. He was really testing your ability to remember, specifically in this case, nonsense words. But it was just a very simple memory experiment showing that we have this decay in memory over time, but you are still able to retain information even a month later, and then people have done studies now years later. So then we jump into a couple, I don't know how many years ahead this is, but maybe 50 years ahead to the start of behaviorism. So behaviorism was a very prevalent area that people started getting into in psychology at this time and really focused on learning. So we're going to talk about the contrast between that and what we study now, or that we study both learning and other things now, but the behaviorists were really focused on learning. So they said, there's sort of nothing innate everything that, all your behaviors and all your thoughts are learned throughout your lifetime. So they wanted, their goals were really to predict and control behavior through different types of learning. And then they really weren't concerned with what's happening in between. They weren't concerned with what we would call mental processes. They just wanted, to show a stimulus and see what your response is to that stimulus. And then modify behavior through learning and associating that stimulus to response. So they really rejected concepts of the mind, of imagery, of thinking, of consciousness, all these things that we're really interested in now. They didn't care about that. They just want to say, okay, if I clap my hands, we should have all looked over here. You're not very good at getting conditioned. Maybe I'm not close enough to you. So I'm sure you've seen some of these famous experiments. There's poor little Albert was a little baby that was studied for a long time on his conditioning. They'd clap really loud next to his head, and he would turn his head. Over time, you learn that. Over time, you may actually, if you're not really getting a shock or anything else, then you're going to start habituating. You stop looking towards it. But these are the types of experiments that they did, the behaviorists. So they're really looking at a conditioned stimulus, and I'll show just a quick example of that. But all this is, they're really just talking about learned behavior. They're doing something and training you to behave a particular way without caring too much about what's happening in between. So what's happening in your mind, or what are the mental processes that lead to that behavior? So of course, the famous cases of behaviorism are the people doing different types of conditioning. So classical conditioning, which is the one I just did. And then there's also instrumental learning. So classical conditioning is just what I said of associating a stimulus with response. Instrumental learning is more of looking for a goal or an outcome. So these are the Skinner box, where they put cats in a box and they had to push a lever that would get them released from the box or give them a reward. So in that case, you're really seeking a reward in order to reinforce that behavior. And so, again, the important point here is that they're only really associating a stimulus with a response, and they're not really caring about what's happening in between. They're really focused on that pattern of learning. So I'm sure all of you are how many of you are not familiar with Pavlov? So I have a little video, but... Oh, I have plenty of time, then. I guess I'll show it. If it'll work. Let's see if it'll work.
Behavioral psychologists have come up with new views, not only of animal behavior, but of human nature as well. And these views all concern a process that we take for granted, learning, because we are all truly born to learn. Ironically, one of the most important figures in the study of learning, Ivan Pavlov, wasn't concerned with the subject at all, at least not at first. Pavlov, a noted Russian scientist, won the Nobel Prize for Physiology and Medicine in 1904. As this original footage shows, Pavlov was initially interested in digestion and the action of the salivary glands. By diverting the saliva of dogs into test tubes, he could precisely measure if and how much they salivated during digestion. When food was presented, the dog salivated quickly, an inherited salivary reflex. But over repeated testings, a strange thing happened. The dog salivated before contact with the food. Just the sight of the food was enough to stimulate their drooling. Then, just seeing the food dish, or even hearing the footsteps of Pavlov or his assistants, was enough to trigger this built-in reflex. What was going on to elicit this response? Pavlov decided to find out by systematically varying the stimuli and measuring the dog's reaction. Metronomes, lights, and bells were all used as stimuli, and they all worked as stand-ins for the food. What mattered was not the kind of stimulus that was used, but the fact that it reliably signaled that food was on the way. Pavlov had discovered a fundamental type of learning called classical conditioning. An original stimulus elicits an automatic, unlearned response. Both stimulus and response happen naturally. They are unconditioned. Then a second, neutral stimulus that never elicits the unconditioned response by itself is introduced just before the presentation of the original stimulus. If the neutral or signaling stimulus is presented alone, and a response occurs as if the original stimulus were still there, we say that conditioning has taken place. The arbitrary neutral stimulus becomes a conditioned stimulus. The reverse is also true. Pavlov and others studied the extinction over time of such conditioned responses. When the subject learns that the conditioned stimulus no longer signals a desired event, the acquisition process is reversed, as the learned connection is gradually weakened. Pavlov's work, and the work of those who followed him, led to a remarkable conclusion. And that is, any stimulus an organism can perceive is capable of eliciting any reaction the organism is capable of making. This means that virtually any sound, sight, or smell can influence the way our muscles tense or relax, our moods fluctuate, or even the way our attitudes are formed. For instance, if I say, relax, and then do this, you're going to be startled and upset. After 5 or 6 pairings of relax, just saying the word relax is going to generate a negative response rather than its usual learned reaction.
All right, so that is one type of conditioning. There's also another type of conditioning, instrumental learning, which is more about seeking rewards. I won't show that video. It's just a very simple video of a mouse pressing a lever to get a reward in a cage. So there's different types of conditioning that happen. But again, a lot of the research in behaviorism is really focused on the stimulus and association with a response, with not really caring about what's happening in between. So what's going on? What are the mental processes that result in this relationship between the stimulus and the response? But there's a lot that we, in cognitive neuroscience, we're very interested in those mental processes. So it's been a change from the traditional behaviorist approach. But there's a lot that we have gained from the rigorous methods of behaviorism. So behaviorism was sort of the first area in psychology that used very rigorous experimental paradigms. So we've taken that and developed that into these methods for cognitive neuroscience, and also objectivity in research. So we're not just taking self-reports or people describing how they're feeling. We're actually looking at an experimental situation where you can manipulate and control conditions and looking at some measurable response, whether that be in the case of these conditioning paradigms, measuring how long the dog was drooling for or something like that, or in humans we're often looking at reaction times or their responses to particular conditions. So these are the experimental approaches we use now are really strongly motivated by the rigorous experimentation that was done by the behaviorist. But there was a cognitive revolution at some point. So this little Far Side cartoon that says, stimulus, response, don't you ever think? So yeah, right. So this is where the cognitive psychologists came in the 50s and 60s and said, what's going on in between? So you have the stimulus and you can measure the response, but what's going on in the mind that's creating this association between the stimulus and response? It also allowed us to explore areas that are not such simplistic conditions. So we can look at things like consciousness and memory and higher level abilities that are harder to study in the behaviorist approach with animals. So that's where the cognitive revolution came along. So there's a stimulus, and we're still looking at the response. That's a measurable response, but we're really looking now deeper into what's happening in the mental processes. And so this is where cognitive psychology really came from, this idea of studying those mental processes that happen in between the stimulus and response. And looking at both the stimulus and the response, but also inferring on what's happening in between. So how do we do that? So one thing that happens sort of simultaneously with this development of cognitive psychology was the development of computers, which I'll talk about in a minute. But just to talk about a few of the proponents of the cognitive revolution, Noam Chomsky, who is still an active researcher. I ran into him. I went to visit a friend at MIT, and there he was in the cafe. Being a scientist that I am, it was so exciting to see Noam Chomsky eating a soup at lunch. I did not approach him and ask for an autograph. I should have. But he really challenged behaviorist views. Specifically, his area of research is in human language, although he's also well known for a lot of his political involvement as well. So he's kind of got these two areas that he's interested in. But back in the 50s and 60s, Noam Chomsky was one of the proponents saying, not all behavior is learned. So there are things, if you look at different animals, even as as infants, they behave differently. So that can't possibly be learned. It's happening too early on. So there are innate behaviors. Specifically in human language, Noam Chomsky argues that every word that I generate or sentence that I generate is novel. So it's not possible that somebody taught me this particular sentence that I'm saying. I'm generating that new. So there must be some innate structure to language. There's also similar grammar or structure of language or syntax across different languages. Why would that be? Well, it's probably not learned because we haven't been exposed to all those languages, but there must be some innate characteristics built into the brain that make you develop language in a similar way. And then there was another, I'm forgetting the author of this, but The Misbehavior of Organisms was another book that was written talking about these differences in behavior patterns between different species, so that not everything is learned. There are some innate differences that happen in different organisms. And both of these are really critiques of the behaviorist view, because the behaviorist view would say that everything is about learning, right? You have to associate a stimulus with response, and over time, you develop this certain behavior. Given a stimulus, you respond in a particular way. And Noam Chomsky and others in the cognitive revolution said, well, not necessarily. There are behaviors that are innate, that you're born with, and you're going to behave differently than another organism just by the fact that your brain was developed in a different way. So then, as I said, the cognitive revolution really came along at the time that computers were being developed. And so a lot of early cognitive psychology experiments and areas of cognitive psychology developed around the idea that the brain or the mind is working like a computer. So there's all these models, and this is still very prevalent within cognitive psychology, all these box and arrow models that are very reminiscent of thinking about the way that a computer processes information. So this is just one example. This is a simple model of attention. So when you're attending to information, I'm only attending to one piece of information at a time. I try to filter out irrelevant information. So in the domain of vision, it's thought of as you sort of have your input channels that go to your senses. So for example, your eye. And then some of that is maintained in a short-term memory store. Memory store is exactly the terminology that would be used in computer language. And then that information is filtered out or selected, and that's what you're actually thinking about, or that's what's leading up to filtering irrelevant information, only focusing on relevant information. So a lot of these box and arrow diagrams are still prevalent in cognitive psychology to explain different cognitive processes, in this case attention, but we'll talk about other ones as well. But again, the important point here is we're thinking about what's going on. So the behaviorist would just talk about the input and the output and not focus on sort of how that's happening in between. And the cognitive psychologist said, well, we want to know what's happening with, between the stimulus and the response, and not just about learning. So the current paradigms in cognitive psychology is really dependent on this information processing, which comes from the computer revolution. So it's really the most prevalent way to think about cognition, and it views the mind as a computer and uses these box and arrow diagrams. So a lot of cognitive psychology, if you read a cognitive psychology textbook, you would see loads of these. So every type of cognition, they have some box and arrow diagrams that explain how information flows through that system. But they're not talking about the brain. So that's cognitive psychology. They're kind of not thinking about this in the context of how does the brain actually compute those processes. So that's where it becomes important to start thinking about neuroscience. So the cognitive neuroscience, as opposed to cognitive psychology, is really trying to take those models from cognitive psychology, your box and arrow diagrams, breaking down these cognitive processes, and seeing how that is actually implemented in the brain, or if it's not actually implemented in the brain in the way that you predict. So it helps guide and interpret the neuroscience results. So cognitive theories really take complex cognition and break it down into its parts, like we saw with attention. There's different stages of attention. So it can really, those models can really guide what you're studying in neuroscience. So in neuroscience, there's billions of neurons. If you just jump into the brain and look at what's going on, it's really hard to think about those results unless you have some sort of cognitive model to interpret those results. And then it really helps to break down those processes into its component parts. So I have this debate a lot with my students. I have a lot of students that are coming from mostly neuroscience. And if you don't talk about a molecule, they'll say, well, this isn't science. Why do we care about this? And I say, well, you don't know what the molecule is doing unless you have a model. And a lot of those models come from cognitive psychology in the case of cognitive neuroscience. And then, obviously I'm kind of in between these two fields, so I obviously think that neuroscience evidence is very important as well. So the opposite spectrum is you have the psychology students who say, well, why do we need to study neuroscience? We've already created these models from psychology. We can see people's behavior. We can create these models. And I say, well, we can create any models we want. How do we know that's actually how we do it? So there have been historically, people have come up with very famous cognitive models that were around for years and were believed to be true until we found evidence from neuroscience that the brain actually doesn't do it in the way that we thought it did. So our brain has developed over evolution. It may not do things in exactly the way that we would think would be most efficient. So neuroscience can really help constrain those cognitive models and modify those cognitive models and inform cognitive theory. And it tells us how the brain actually does it. So not just what we've decided makes the most sense at a cognitive level, but how it actually performs those computations. So that's sort of the first part, the history of cognitive neuroscience. Are there any questions on that part? And feel free throughout, if you have any questions, to just jump in. Go ahead.
This is not real important. I just wanted to mention that was Phil Zimbardo introducing the whole Pavlov thing. We watched those videos when I took Psych 101 in 1990.
Oh, yeah, yeah. Don't tell my students that. Yeah, no.
Then we learned about the Stanford prison experiment, and then they got funnier.
Right.
Okay, thank you.
I may be jumping ahead, but is imaging showing us that things are happening, the processes are happening in ways that we didn't predict, or that there's other things going on at the same time that aren't easily observable?
Yes. I don't have a particular example here, but I do in my intro cog neuro class. I do talk about why would we care about this neuroscience evidence? There are a few particularly famous cases in which there were competing researchers in the field of cognitive psychology had their own sort of pet models, and they for a long time studied each, and they would find behavioral evidence that would not disambiguate those models. So you find evidence for one or you find evidence for the other, but we couldn't never really tell which one was right. And that's where the neuroscience evidence came in and has been able to dissociate those two and say, okay, the neuroscience evidence suggests that it's one model and not the other model. So that's where it really can help provide evidence either contrary to a model or in support of a model and help modify those cognitive theories. Other questions? I will talk about neuroimaging evidence, or some neuroimaging studies later on, though. All right, so again, I'm going to just give you, in this sort of second part here, I'll give you a broad overview of the types of things that people are studying in cognitive neuroscience. Now, I am not an expert in all of these areas. I've learned about them through coursework and in graduate school and have worked with labs that have done some of this research. But my real focus is executive function and memory. So they're two components of what we study in cognitive neuroscience. And I'll get into a little more detail on those particular ones. And the examples I have later on are all about memory. But the cognitive neuroscience in general studies domains, all of thinking, which I'm probably not even including all of them here, but perception, attention, memory, emotion, and how emotion affects other types of cognition, the development of language, executive function, and decision making. So if you don't know what executive function is, that's really sort of what makes us unique as humans, is we are sort of self-reflective. We can control our behaviors. We're not just acting like our dog was in getting conditioned to do behaviors, but we have this ability to control our behaviors and modify our behaviors to reach some sort of goal. So that's what executive function does. And executive function and emotion really interact with all of these other types of cognition. So none of these are sort of happening on their own. They're sort of all interacting to create behavior. So I'll just go through a couple examples of each of these areas. So in perception, the types of things people study are how do we process information and perceive through our sensory systems, how do we perceive information. So one area is through vision. So I'm only going to talk about vision, but obviously this applies to other senses as well. There's a whole field of research in auditory and cognitive neuroscience. But for vision, one of the areas that people do research on is how do we process information about faces differently than seeing other types of objects. And this is one of those studies. So if you show a bunch of pictures, these are pictures of faces and then heavily pixelated images. It will also be the case if you just showed a picture of a face versus, say, a picture of I don't know, a clock or some other type of object that doesn't have a face. I know clock has a face. That was a bad example. Well, the problem, you can't ever use, this is actually a real problem in this field of research, because if you use pictures of other things, like people have tried to do studies of comparing faces to birds, but birds have faces too. So not the same, and we're probably not adapted to seeing bird faces as much as human faces, but a lot of things have characteristic features of a face, even cars. If you show people a head like the front of a car, it kind of has face characteristics. So you see some of the same activity happening for those. Anyway, it's beyond the scope of what we'll talk about. But if you look at the activity in the brain happening for faces compared to other objects, you get a lot of activity, specifically in right. And I know I'm pointing to the left. This is radiological convention. It's like you're looking at the head below. So it's flipped, but it's right. It's called a fusiform gyrus. So this is the area where I'm sure you've heard, like in the news, I don't know, maybe 5 to 10 years ago now, the Halle Berry neuron. This is not. No cognitive neuroscience really believes that that's truly the case, that you have one neuron for every face you've ever seen. But this area will be highly active for faces compared to other types of objects, and that's pretty consistent through the research. More interestingly, in recent years, people have said, okay, well, maybe it's not just about faces. Maybe we're just really good at looking at faces. We've been, when you're a baby, people put their face right in you're seeing faces. You're getting a lot of exposure to faces. And maybe this area is adapted to just anything you've had a lot of exposure to, or you're an expert at identifying faces. And this is actually true. So if you look at people who are experts in identifying cars or identifying birds, you see activity in the same area. So if you get a car expert, you'll have higher activity for cars than other objects. It's not quite to the level of faces, but you're still quite good at it, and you'll show the activity pattern that's higher compared to, if you're a bird expert, you'll have higher activity for birds. So it's thought that this area is really an area not necessarily about face processing, but areas of expertise, visual expertise specifically. There's also, of course, there's other areas in the brain that are active for all sorts of types of visual processing, up from the lowest level of just processing light and light-dark gradients all the way up to objects, like we see in the face processing areas, as well as other types of objects that are not faces. So these are, researchers are doing a lot of study at all of those levels, from the very simple, light-dark gradients all the way up to processing more complex objects and visual features. And I should also note that within all of these areas, people are also doing research on what happens if you have brain damage to these regions. So there are people in perception or in face processing that have a disorder called prosopagnosia, which is damage to this fusiform area, specifically right fusiform gyrus, and they have an inability to recognize faces. And then, so the next one I'll talk quickly about is attention. So who is familiar with this study? Probably too many of you to bother showing it then. So if you've already seen this, you will be ruined for life. You will never. So I'm gonna ruin it now for you. I'll show you the video, but I'll kind of talk about it as it's going on. But the idea with attention research is there's many types of different attention research, but one of the areas is looking at selective attention and our ability to really focus attention on one thing. And we often think of that as a good thing. If I'm studying for a test, I really want to filter out all the noise surrounding me. I want to focus on, if I'm driving, I want to focus on the road ahead of me. I want, not pay attention to all the distractors happening on around me. There's also divided attention, so what happens when you're dividing your attention between multiple things. So there's a lot of research that goes into all these different types of cognition. I'm not showing here an image of the brain, but there are areas of the parietal cortex. Oh, I wish I could draw my brain. I don't really have space for it. You can see down here, your parietal cortex is about here, very large area. Where I was just talking about is basically here. That's a fusiform. This is occipital, which is all vision. And then this is attention, as well as other things. Again, these are very simplistic. There's a lot going on in all of these regions. And this visual processing sort of happens along this stream. And then you get a lot of spatial attention and attentional guiding of vision that's happening from these areas of parietal cortex. What happens in this video is a demonstration of selective attention. So if I ask you to pay attention to the people in the white shirts, then they're going to be passing a basketball to each other. And I want you to count the number of times that they pass the basketball. I play the video. You would count the number of times. And then I would later ask you, so did you see anything unusual here? And if you're really good at selective attention, you didn't notice anything else because you've done a very good job of selecting out just the information that's relevant, the passing of the balls, and not paying attention to anything else. But what happens is there's a guy in a big gorilla suit that walks across, counts his chest, and continues on. And a lot of people will not notice that because they're focused on the passing of the ball. But when you watch it again and know that that's there, it's very funny that you completely miss this very obvious thing. So, people think of this as oh gosh, I didn't see that really obvious thing, but it's an advantage of our selective attention system that we can really filter out irrelevant information and focus on what we're intending to focus on. So now you can go online and find this video if you want to see what it looks like. But I've ruined it. Now that you know the girl is there, it's very hard to not pay attention to it.
Is there a named disorder for a lack of specific attention?
Oh, there's different types of attentional problems. So there can be unilateral neglect, as if you have damage to one side of parietal cortex. Generally, it tends to be the right side of parietal cortex is really involved in spatial attention. So you can have neglect where it's not that the person cannot see what's on the left side of space, but they won't attend to it. So somebody with this type of, it happens a lot with certain types of stroke. If you have this unilateral neglect, what will happen is these patients will only eat stuff that's on the right side of their plate. They'll only button their shirt on the right side. They'll do all these things that indicate that they're really only attending to the right side of space. It's not that they can't do it, so they can see still. So if you actually draw their attention to the other side of space, what they were ignoring in the first place, they will be able to process and attend to, but they won't sort of naturally guide their attention to the opposite side of space. But yeah, that's an important distinction to make. There's a difference between just not being able to see, and if that would be damage to the occipital cortex, versus not attending to that information, which is generally driven by lateral parietal cortex. This is my example. One of my favorite movies, of course, because I study memory, is Memento. It's not accurate, but it's pretty good. It's interesting. So my area of research is memory, and that's a very broad area. So memory is really involved in sort of everything. Any learning is memory, so it's involved in kind of all areas of cognition. But specifically, I'm interested in how we both encode information, so how we learn information, and then how we retrieve it later on. So a lot of my research involves showing people either words or pictures, having them study these lists of items, and later asking them to retrieve that information and seeing what happens in the brain as they're retrieving that information. And so I'll show you a couple examples of my own research later on, but we see a very broad area of activity within the brain for memory. And I'll talk about this as sort of in the current directions. It's not that any single brain area is working on its own at any one point in time, and I think a lot of this misconception comes from a lot of neuroscience research showing activity, and when I showed you the fusiform gyre, so it looks like the rest of the brain is doing nothing. And the important thing to remember is it's always compared to some sort of baseline. So it's not like your brain is shut off in every other area while you're doing that. It's just that it's always active in the other condition, or your baseline condition. So I'm sure you've all heard of this myth of the 10% of your brain is only active at any one time. That is not true. Your brain is firing, all areas of your brain are firing all the time. And so in memory, I'll show you later on an MRI image from my own research, but there's really not one single area that's involved in memory. It's a large network of activity in frontal cortex. This is temporal regions. Oh, I should have oriented you to my brain. Here's your eyeball. So it's not really anatomically correct, but that's where your eyeball would be. This would be the back of your brain. And this is like the side, so it would be taken like a slice through the, so it would be like if my head were like this, going this way. But what you can't see is in the inside of there is a thing called the hippocampus. And if you've taken Latin, you might know, oh, that's a really terrible writing, sorry. Hippocampus, if you know any Latin, that's actually seahorse, and it's kind of shaped like a seahorse. So that's kind of within, that's right in the medial part of the brain, so right in the middle. So it's like I took a slice out of the brain like this, right down the center. And so that area is sort of historically been thought to be very important for memory, but what we know now is, hippocampus isn't working on its own. It's heavily influenced by frontal cortex, and as well as temporal areas, lateral temporal areas, so outside of the middle of the brain, as well as it's guided by attention. So all of these different areas are really interacting. That's what I like to show an MRI image of memory and say, you thought that it was just the hippocampus, but look, I give somebody something to remember, and their whole brain lights up. So it's not just the hippocampus, it's all of these areas interacting. And then, of course, in the study of memory, we also look at people like our patients with disorders, so particularly Alzheimer's disease. There's a lot of research on people with damage. So a lot of the research showing that hippocampus is important for memory comes from patient HM, who was treated for epilepsy, incurable epilepsy, and they took out his hippocampus, and lo and behold, he couldn't remember. He couldn't encode new information. So we learned a lot about memory by that unfortunate surgery that was done. He was sort of desperate to get rid of his symptoms of epilepsy, and in order to cure that, they took out the tissue that was causing epilepsy, but in the process of doing that, they really ruined his memory system. Go ahead.
Do you look at any books to self-identify eidetic memories, photographic memories?
I don't. There's not, actually, surprisingly, there's not a whole lot of research on it. I have done, personally I have not done research actually with those either patient populations or people with superior memory, but there is some research now. So there's somebody, I'm forgetting his name, but he's at UC Irvine, that's doing a lot of research now on people that have sort of perfect memory or photographic memory. Now I'm a little skeptical of that because the people that I've at least seen some video of are also tend to be, so there's one particular woman that I've seen some research on that she does seem to have an enlarged hippocampus, which she may have been born with, which indicates she would have some better memory abilities. But she also tends to be somebody who obsessively writes down everything that happens during her day. So it's not really clear to me that it's sort of an innate ability or there's the fact that she's really practicing a lot, right? If I learn an instrument, if I practice a lot, I'm going to get really good at it. It's the same thing. I think she's practicing memorizing things a lot. So it's not really clear to me yet that the research hasn't concluded whether there really truly is such a thing as sort of innate perfect memory ability. But yes, there's in all of these areas, and I'll talk about this at the end, there's individual differences. I like to say that my theory is that people who are psychologists study psychology because they want to understand something that's wrong with them. I have terrible memory, and that's why I want to understand it. Although I've gotten better because I have learned a lot of tricks from my research. And so another area that cognitive neuroscientists study is emotion. So this is also probably from your Psych 101 class. This is, what is his name? Paul Ekman, who studied different emotional responses, and he showed these images across different cultures and shows that there's sort of basic emotional facial expressions that are consistent across cultures, which indicates, again, that it's maybe an innate process. There are variations across cultures as well, but there are a subset of those that seem to be consistent across all people. But we, in cognitive neuroscience, also want to understand how emotion develops, sort of what its purpose is, and how it affects other types of cognition. So a lot of the research in emotion and cognitive neuroscience is really how emotion influences other types of cognition. So we talk a lot in my memory seminar about how emotion actually amplifies signal within the hippocampus. So if something's emotional, you tend to encode it more. So you're going to be better memory if you have an emotional stimuli. It can also affect other types of cognition, like decision-making, things like that. So it's not only do we want to understand emotion in and of itself, but we also want to understand how it affects other types of cognition. And as I've been trying to think of a disorder that goes with each of these, which I do in my course as well, I always end my lectures with some disorder. People with autism have some differences in the way they process emotions, so they won't. So when we look at faces like this, we tend to focus on the eyes to get a lot of the emotional content. And some people with autism don't focus so much. Their eye tracking, if you were to track where their eyes are looking, they're not really focusing so much on the areas that convey that emotional information. So they have a deficit in processing that emotional information. And so in language, there's, again, a whole other area of research. People are looking at what areas of the brain process language information, how that develops over the course of your life, how do you learn to process different languages. My example here is just an example of how what you see contributes to how you perceive language. So if this works, I'll play this real quick. So close your eyes first, and I'll play this video. There might be an advertisement comes up first. No, there isn't. Good.
Ba, 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 ba.
All right, so what do you think he was saying? Okay, now, just go back to, so now keep your eyes open and see what he's saying.
Ba, 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 ba.
Yeah, so as you can see, when you see his mouth moving, so what he's saying, what his mouth is doing, sorry, I've opened the wrong thing here. What his mouth is moving and what he's saying are ambiguous, so what he's saying is ambiguous, but by seeing how his mouth is moving, that constrains how you perceive that sound. So when we think of language, it's not really just happening through the auditory system. It's also constrained by body language, by the way he's moving his mouth. There's a lot of processing of language information that's very ambiguous. If you were just to take a spectrogram of what that sound looks like, it's very hard to tell the difference between when words cut off, especially for me because I talk so fast. So there's a lot of information we're getting from the visual system and other systems that constrains how we're perceiving language. So in cognitive neuroscience, there's a whole slew, again, of research in both language development. There's also cross-cultural studies in looking at how different bilingual people's cognition is different than a monolingual person, the development of bilingualism, and areas of the brain that are active or more active when you're bilingual than monolingual. There's all sorts of research on the development of language from childhood through adulthood. So this is just one piece, but what we have learned is that language is sort of not acting, our auditory system is not developing language on its own. It's really constrained by these other cognitive systems. Another area of research, which is one area that I study pretty heavily, is the executive function. So who knows who this is? Yes, Phineas Gage. So Phineas Gage is, again, just like patient HM, we learned a lot about cognition from these patients who had these unfortunate accidents, or in the case of Phineas Gage, he was working on the railways, and he was using a tamping iron to put dynamite into rocks to blow them up, and unfortunately it ignited, and the tamping iron flew through his head, out his skull, and landed, I don't know, 30 feet away or something like that. Surprisingly, he actually walked away from the scene, disoriented, and his friends took him to the hospital to see a doctor who sewed up his head. At the time, they really didn't know what could be wrong with him. As he lost a significant portion of his frontal cortex, which we have learned a lot about behavior from what happened to Phineas Gage. Now, there's a lot of controversy. There's a lot of debate about how good the recording of his behavior was. So some people think it was sort of sensationalized a little bit. But what we have learned from Phineas Gage is that the frontal cortex, you can live without a frontal cortex. But it tends to guide and constrain your behavior in a way that's consistent with your goals. So Phineas Gage is described as being sort of erratic and impulsive after he had this brain damage. So those frontal cortex, I like to, in my class, I didn't put it in here, it always gets a good laugh, is babies act in a lot of ways like Phineas Gage did, right? So small children don't have a fully developed prefrontal cortex until they're actually, till about 25, your prefrontal cortex is not fully developed. So, people children, including our own college students, tend to act a little bit more irrationally, and it's sometimes hard to understand their behavior, but it's partially because they don't have sort of the level of executive control that we have from a fully developed prefrontal cortex. So I study executive function in the context of memory. So what I want to understand is how the prefrontal cortex guides how you remember information. So it tends to be the case that when you have very difficult pieces of information to remember, or there's a lot of interfering information, the frontal cortex basically jumps in and says, okay, you should focus on this piece and ignore irrelevant information. It guides how you remember information. So you can think of it as like sort of a support process of guiding these other types of cognition. And then finally, decision making. This is a big area of research, and a lot of this area of research is focusing on how we make decisions in certain types of contexts, specifically looking at rewards, so how we make decisions that will lead to reward. Obviously, in humans, that can be very complicated. You know, you go to graduate school expecting some long-term reward, which is happening, you know, years in the future, and it's very hard to predict what that reward is going to be. But a lot of our decisions are guided by our expectation that we will be rewarded for our behavior. Now, this is an area where I like to talk about how this relates to other fields, because this is an area in cognitive neuroscience that has been really popular lately among behavioral economists. So they've taken these ideas from cognitive neuroscience and just run with it, with trying to predict people's behavior in complex, you know, marketing situations. So, and this is a really big hot topic among students these days. I had, I think, 2 or 3 students go on to work for neuromarketing companies this past year, and these companies are using neuroimaging, different methods from cognitive neuroscience, using that information to try to predict and, you know, in a way, control our behavior. Try to predict what we're going to buy based on the information that they're showing you in their advertisements is one of the areas where they're using neuromarketing. And I actually have there might be a reason why I have 3 students doing this, is because I have a good friend from graduate school that started her own startup neuromarketing company based on her dissertation research. So she looked at showing people visual images of objects. So say you know, do you feel positive or negative about this object? Most things we think of are as very neutral. We still our brain will respond in a way that's consistent with it being positive or negative. She calls this microvalence, so you're either going to have sort of a slightly positive or slightly negative response to this. So what she's done for years, marketers have used, you know, bring people into a lab, they show them images or show them different advertisements, and they say, you know, do you like this part of the video? Do you not like that part of the video? And they, you know, over years they modify their advertisements in accordance with people's preferences. Well, you don't need to do that anymore. She's taken the brain activity and said, I can predict based on brain activity what videos people are going to like, or what images people are going to like or not like, and she can feed this through a computer program that will give you that prediction without ever having to bring a person in to watch these videos. So she's taken her evidence from neuroimaging and really, you know, made that into a new way to do marketing, and through using the evidence from neuroimaging. If you're interested in that, it's called Neon Lab. It's pretty neat. And so these are all areas of cognitive neuroscience that, you know, each one of these areas are hundreds of researchers doing research in these areas, and I've only given you, you know, a small taste of each of these areas. But my research is specifically on memory, and then I also have done some research on executive function. So the next part here is I just want to give you a little bit of a taste of these different methods in cognitive neuroscience. So there are lots of methods, and I'm going to talk about a few that I am the most comfortable with. Those are the methods that I have used, but I'll just mention quickly a couple of other ones as well. So in cognitive neuroscience, there are many different types of methods. People often will combine different types of methods. That's one of the future directions, I would say, is that it's very hard now to get funding if you only use one method. You really want to combine across methods and show converging evidence. But from cognitive psychology, we've taken, again, a lot of the experimental methods that have come out of behaviorism and then the cognitive revolution. So we use behavioral methods that are very similar. I mean, if you come into my lab, you're doing a task that would be almost identical to what you would do in a psychology lab. It's just that I've also got you hooked up to this machine that's going to look at your neural activity. So you have these behavioral methods that are done. And then really what we want to know is we're localizing brain function or looking at other neural activity. So localizing brain function can be done through numerous neuroimaging methods, but it can also be done, which I won't talk about too much here, but with patients with lesions. So if I know that you have a lesion in a particular area of your brain and you can't perform a specific behavior, then I can associate that region with that behavior. We can do that sort of in people who already have lesions. Obviously, in humans, it's not ethical to give them lesions. In animals, you can give them lesions and look at brain functioning in those areas. But of course, it's always helpful to have sort of both animal research and human research that provides converging evidence. And then what we can also do that is more sort of recent development in human research is brain perturbation methods. So this is, again, done in animals and humans. But there are ways that you can sort of temporarily disrupt activity in certain brain regions. So there's something called transcranial magnetic stimulation. And if you stimulate at certain frequencies, you can either enhance processing, increase neural activity, or you can suppress it. So it's kind of you can think of it as a temporary lesion. I can sort of shut that area of your brain off temporarily, look at how that affects your behavior, but by the time you leave the lab, you're back to normal and safe. And there's other types of methods that look at measuring neural activity. So again, there's a distinction here between actually invasively going in and lesioning, or even using non-invasive brain perturbation methods, and then looking at just the correlation of activity in a certain task and measuring that neural activity with some sort of neuroimaging method. And I'll mostly be talking about measuring neural activity at the bottom here, but then I'll talk a little bit about some future directions in using brain perturbation methods. So this is sort of a general overview of cognitive neuroscience methods. And what we always talk about in cognitive neuroscience is that there's no perfect method. There's two sort of dimensions that we're most interested in improving our methods on. So the first is temporal resolution, so how quick that activity occurs is on the x-axis here. So our brain is processing things really fast. Neurons are firing really rapidly. So what we want is really down to the millisecond level. That is very fast. So our ideal method would be down to 1 millisecond or 10 milliseconds at the most. And then on the y-axis here is the spatial resolution, which is how small of an area of the brain are we looking at. So we've got on the, oh gosh, is that micrometers up to a meter. So obviously, the top there is looking at the whole head down to individual neurons at the bottom. So all of these methods listed up here fall somewhere within this temporal resolution and spatial resolution. The two I'm going to talk about is fMRI, that big yellow one up there. Very slow temporal resolution, relatively good compared to other cognitive neuroscience methods that can be used in humans for spatial resolution. So you can get down to a couple millimeters, which is still thousands of neurons, but still a small area of the brain. I'll also talk about EEG, which is up there just at the top of the fMRI methods, and then scalp ERPs, which is over here. Those have very good temporal resolution. We can get down to milliseconds. The electrical activity at the scalp can be detected within a millisecond. But you're getting activity across the whole head, coming from all areas of the brain. And the way that electrical activity spreads at your scalp doesn't give us a very good ability to detect where it's coming from in the brain. And so I'm really going to focus on ERP here and the fMRI techniques, and I'll give you a little bit of background on what that is actually measuring and sort of what we use those to study. But there's, of course, all of these other methods. So the patch clamp recording, single unit recording, those are procedures done in animals. They're invasive. You're actually driving an electrode right into the brain. And then there's other types of optical imaging techniques and then lesion techniques that are used in humans, but many of these are used in both animals and in humans.
Is there no point to having a small-scale, long-term method?
So on the
Big white gap.
so if it were down here, you mean?
Yeah.
Well, I mean, really what you want is something that would be here. So you're trying to maximize both the spatial resolution and the temporal resolution. There's just no method that it's not that we're trying to avoid that area. It's just that there is no method that sort of has a very good spatial resolution but a poor temporal resolution. Yeah. So, I mean, really, you ideally want the best of all of them, and which in this case is the single unit recording, the patch clamp, but that's invasive, so you can't do that in humans. So it's not that these methods do exist, they're just invasive and we can't be done in humans. So it's always nice if what you find at the level of human research corresponds to something that you can do in animals, or you can confirm it with an animal study. So I'm going to specifically focus on EEG, which is electroencephalography. And as you can tell from the name, it's about electrical activity. So it measures the electrical activity that's happening at your scalp. If you want to come see what that looks like, we've got a setup over in our lab at the basement of Canbar Hall. We put electrodes on people. Looks like a swim cap. I'll show it to you in the next slide what the setup looks like. And then what we are able to do is detect electrical activity that's coming from neurons firing in your brain at the level of your scalp. And again, this one has really good temporal resolution, but unfortunately poor spatial resolution. And I'll talk about why that is as I explain these methods. So for both EEG and fMRI, I'm going to go into some details of what exactly is happening in the brain and what we're detecting with these methods. Please stop me if it's not clear. I am not a physicist, so there's some level that I don't understand this stuff either, but I can give you a rough overview of these different techniques. So in an EEG system, this is what our EEG cap looks like. Those are the little electrodes. Each one of those is connected to an amplifier. The signal we're getting is very small. We're amplifying that signal so we can actually see it. And this is what a trace would look like. Sorry, it's probably hard to see there, but it just essentially looks like a wiggly line. There's not a lot you can see on the raw data. Each one of these is an individual channel. That's each electrode that's on the scalp, giving that activity. These ones down here are very large because it's actually eyeball movement. So unfortunately with EEG, one of the big challenges is that any movement or even electrical noise within the room will cause a lot of interference to the signal, because what we're detecting are very, very, very small effects relative to really big effects that you get with noise. So we're looking in the case of in my lab, which this cap is too, there's 64 channels for 64 electrodes on the scalp that we can record this electrical activity. And this is continuous. So this is just showing a screenshot of sort of one point in time. But each of these little segments here is a second. And that's sort of continually going for an hour-long experiment. We get a lot of data. It's very rich, large data sets. I noticed one of the sort of suggested points of discussion is how we handle data. This is a big challenge in cognitive neuroscience. Our data is large. We take up a lot of server space. And it's also very challenging to sort of interact with other researchers doing your research, and being able to share data is a big issue because this data set is so big. So this is what our setup looks like. Sorry,
I'm sorry,
go ahead.
Curious if you could give me an idea of what like a grand mal seizure would look like on in relation to that baseline.
You would see it, yeah, you would see much larger amplitude spikes that would happen. So there's certain characteristics of different types of seizures that you would be able to detect. You would be able to detect that in raw data. But a cognitive effect, if my students are always so excited when they start in the lab, they're like watching the first subject, and they're like, can I see when he's remembering the information? You can't see anything from this, but noise. I mean, noise is big. But when you do have, so epilepsy or a seizure is really sort of erratically synchronized activity, which causes that high amplitude. So you would see spikes here that were characteristic of that. Yeah, obviously, I mean, EEG methods were developed for the detection of seizures, and that's very easy to see in raw data. But you're not going to see much happening until you've processed the data if you're looking at a cognitive task. What you can tell is alpha activity, which is a certain 10 hertz frequency. When people are sort of getting very sleepy and they're very bored of your task, you can see that. Or you can see when they're blinking. This down here is eye movement activity. So I'll just quickly go through what actually drives this electrical signal that you can find at the scalp. So if all of this is sort of over your head, just ignore that this is a neuron. Just think of it as a battery. It's happening the same way as a battery. So in a resting state, you have more positive charge or positive ions outside the cell than inside the cell. But when it's active, those positive ions rush into the cell. And what that creates is a dipole, or a separation of those charges inside and outside of the cell. And you'll get basically a battery happening. Was that my 5-minute mark? Oh gosh, I'm way behind. OK, so you'll get sort of this battery happening. So I'm gonna skip through a lot of the rest of the EEG stuff, I think, for the interest of time. But this is basically just taking a separation of those charges, and when you get a lot of neurons synchronously active such that you have that separation of charge across a ton of different neurons, you're going to be able to detect that positive end of the dipole, so you'll get a positive part and a negative part, just like you would in a battery. You can actually detect that at the scalp. So if there's a lot of synchronously active activity in a certain area, that's going to spread out the skull and the scalp, and because it's electrical, it's instantaneous, so that's why we have really good temporal resolution with EEG, and you can detect that activity at the level of these scalp electrodes. That's essentially what the next 5 slides say, so I'm going to skip through those. So ignore all this, ignore all that. Let me go to this. So like I said, with the raw data, it's really hard to see anything coming out of that. But what we get is on every trial, say we're going to show a stimulus onset on every trial, and then we have some activities happening after that. If we take many, many, many trials and average across those, you get this very nice characteristic waveform. Each of these waveforms is related to some sort of cognitive processing. The early ones, the beginning here, is usually visual input or auditory input, so sensory stuff, all the way out to sort of higher level cognitive processing at the other end. So once we take different stimuli across a number of different trials and add those all up or average across them, we can look at separations between conditions. So this is called the error-related negativity. So when you have an error, you have this characteristic spike in activity relative to a condition where you don't have an error. So that's just one example of a particular event-related potential. And at different points in time, there are different types of cognitive operations that we can study. So later out in time is actually memory effects that we can see. So for the interest of time, maybe I'll skip fMRI. No?
We have a total of 14 more minutes, and you've been taking questions as we go.
Okay. All right. I'll go through it quickly too. But so with fMRI, it's very different than what we're measuring with EEG. So with fMRI, what we're measuring is really changes in blood flow. So as you can imagine, it's not quite as fast as detecting instantaneously when a neuron fires. In this case, you're looking at when neurons fire, they need energy. So they're going to need oxygen. So the blood flows to those regions, deposits the oxygen, and it's that difference between oxygenated blood versus deoxygenated blood that is actually detecting that activity in those brain regions. Now, it takes approximately about 6 seconds to get a peak of that blood flow response. So it's not on the matter of milliseconds like you're getting with EEG. It's taking quite a while for that energy, that oxygenated blood to get to those brain regions. But it has quite good spatial resolution. So there's some trade-offs, again, between these different techniques. So with fMRI, you're looking at increased neural activity causing that increase in oxygen to those regions, and it's able to detect the difference between oxygenated and deoxygenated blood, or the ratio of those, to give you an idea of where activity occurs within the brain. So this is an example of, up there is what the fMRI machine looks like. There's a giant magnet that the person has pushed into. Some of you may have had this for knee surgery or something like that. If you put the brain in there, you can get nice images of the of cortical structures and see where activity is occurring by that difference in the magnetic properties of oxygenated versus deoxygenated blood. So when you have more oxygenated blood going to a region, you can detect activity there. I'm going to skip again through how those are actually generated, but it really comes down to just being able to detect the magnetic properties of the oxygenated blood. This is the example I mentioned before. So if I were to have you study a list of words and later asked to remember those words, it's not just the hippocampus, this area we thought is important for memory, that lights up. You have oxygenated blood going to all of these regions. So there's a lot of frontal cortex. The red is where you're getting more activity. There's some areas of temporal cortex as well as parietal cortex. So all of these areas are contributing to memory. It's not just about hippocampus. So this is what I wanted to make sure I didn't miss. So where is the future of cognitive neuroscience? So we've talked about sort of what people are studying currently and what some of the current methods are in cognitive neuroscience. But I think there's some really exciting new areas that people are sort of directing their research to in cognitive neuroscience. So I can tell you the one thing that NIH and NSF actually will not fund anymore is a picture like this, saying these areas are active. Okay, we've had tons of studies on that. 10 years ago, I could have gotten a job at Harvard if I showed a picture like this. Now it's old news, so nobody cares anymore about this modular definition of each area being active for some function. So what people are really focusing on now is looking at things like individual differences and how different people's brains behave differently, and lifestyle factors that can influence cognition, and then really moving beyond this modular definition. So if this area is active for memory, this area is active for attention, and looking at networks. So we all know, and every cognitive neuroscientist knows, that it's not just one brain region that's active for a particular function. There's a whole network. So now we're really interested in how those networks work. And then we're moving from correlational to causal studies. So all of the things that I've talked about so far in EEG and fMRI are correlational. We say we're correlating what's happening, what you're doing in the task, the behavioral task, with some brain activity patterns. But we don't know for sure that those activity patterns are really causal. So what we really want to know is if I can disturb a certain area of the brain, or when I talked about this transcranial magnetic stimulation, actually shut off that area of your brain, is it actually causally influencing that pattern of behavior? And then finally, we, of course, hope that the research we understand, and basic research, has some application, clinical applications as well. So a lot of research now is moving from using cognitive neuroscience methods that are used in normal people to understand cognition and how it goes wrong in patient populations. And also using techniques that have been developed to understand normal brain functioning to actually modify the brain in order to influence cognition. So I'll just give you a couple of examples, again, in the domain of memory of these different approaches. So in the context of individual differences, there's a lot of evidence now that memory is affected by genetics. So it's not just about learning, but there are some different differences in our genetic makeup that people are predisposed to remember information better. It's very complicated. It's not one gene. There's not a memory gene. There's lots of things going on. And you can look at the differences in genetics and see differences in memory abilities. And I did a study a few years ago looking at this, and it's complicated, but you can pull out some differances in genetics that can predict whether people are going to perform better or worse on a memory task. And then, of course, memory is also an area that's a lot of the brain is active in memory tasks. We know there's a lot of areas that are performing memory. So a lot of different types of mental illness and lesions that can happen in the brain will affect memory. So it's not sort of just, people talk about memory disorders as Alzheimer's disease. Well, it's not. People with schizophrenia, with anxiety disorders, epilepsy. I'm trying to think. I just wrote an NIH grant. I really should know this. All of these disorders have memory disabilities as well. So it's not just about Alzheimer's. So if we understand what's happening in normal functioning memory, it can also help aid us in understanding what's going wrong in people affected by disease. So another thing that's really exciting and interesting, some area that I've also gotten really interested in recently, is different lifestyle factors. So even if you're born with these genes that make you remember things badly, is there something I can do to save myself from the inevitable doom of not being able to ever remember anything? Well, there's a couple things you can do. Get good sleep. There's a lot of evidence showing that sleep can heavily influence memory. Your brain is active while you're sleeping. It's consolidating memories. It's replaying those memories while you're sleeping and helping you remember that information better. There's some really exciting new research coming out showing exercise influences on memory, so people are better. Well, there's animal research as well, but there's also studies with people with and without Alzheimer's disease that show improved memory after extended periods of exercise. Don't ask me how much is enough. I don't know. I hope it's like 5 minutes a day, but I don't think so. And then another area I'm really interested in is now I have a question mark on this because there's a lot, mindfulness is like a big thing now, and there's all this popular press saying there's all these effects on cognition, but really there's not a lot of solid research on it. And there's some suggestions from different pieces of information that mindfulness meditation should affect memory, but we don't know that yet, and that's why I'm going to run this study in my lab to see if that's the case. Hopefully there is a difference, but we don't know for sure. So in addition to these sort of future directions, I think that another area is that the types of methods we've used in cognitive neuroscience for understanding normal brain functioning could also be used in the context of therapeutics. So non-invasive brain stimulation is really an interesting new area that people are using for this purpose. So not only can it tell us causal information about how the brain works by disrupting brain activity, but it can also lead to future treatments of patients with disorders. So this is basically my NIH grant summarized in 2 sentences. It's taking non-invasive brain stimulation to change the connectivity patterns in your brain. So basically, if I stimulate your brain over and over and over with this non-invasive brain stimulation, I'm zapping your brain over and over and over, you're going to actually create connections between the frontal cortex and the hippocampus. It's going to enhance those connections. And then by enhancing those connections, it could improve your memory. And this is interesting. Like all of us, I'm excited about this. I want to go get my memory improved. Not only can it help people who are sort of already at the normal level be enhanced, so cognitive enhancement, but it also could potentially be used as a therapeutic treatment for patients with memory disorders. And again, that's not just Alzheimer's. It can be sort of anyone and everyone who has sort of a deficit in this frontal hippocampal network. They get decay of those pathways. You could enhance activity within those pathways and hopefully alleviate some of their memory symptoms. I think that's it.